This is an A-level biology presentation looking at RNA interference. There's a number of terminologies you need to be aware of when talking about DNA and epigenetics. Each of the words and phrases in front of you should be familiar with. Come up with a definition for each one. Pause the presentation, get your answers, and when you're ready you can restart the presentation. Pause now. Well, the answers are as follows. Again, if any of these don't make sense, it would be worth going back and looking over the presentations on epigenetics and the presentations on structure of DNA in order to make sure you know what you're talking about. If you are ready, you can of course continue on with this presentation. Now you've got describe the process of transcription and translation. And there are two main types of answers. You can have a 1 to 2 mark answer, or you can have a 3 or more mark answer. Most of the time these types of questions will be worth 3, 4, 5 or even 6 marks. Have a go now, pause the presentation, come up with your best answer, spend no more than 5 minutes on the whole thing, and when you're ready you can restart the presentation. Pause now. Well, in terms of a 1 or 2 marker, you simply have to say something along the lines of transcription is a process of DNA being used as a template for the production of a messenger RNA strand. You can talk about translation is where a messenger RNA strand and transfer RNAs are used to then produce proteins. For the longer answer questions, of course, you need to go through the processes in a lot more detail, and there are separate presentations looking at transcription and translation that I'd suggest you go back over should you need to. When we're talking about small interfering RNA, we are talking about interfering with the process of translation, which is where the RNA is used as a template and is used in the production of proteins or polypeptides. So where did the double-stranded RNA come from is the first question we need to answer when talking about small interfering RNAs. Well, there's two ways in which we can get double-stranded RNA. The pictures on the left is one, the picture on the right is the other. Pause the presentation, try and make your best guess on how that arrived, and when you're ready, restart the presentation. Well, the picture on the left is where you have DNA is being transcribed into messenger RNA. And what has simply happened is we have two complementary regions of DNA, and as a consequence they produce two messenger RNAs that are also complementary, and in this particular case the two messenger RNAs that are complementary have combined together to form a double-stranded messenger RNA. The other way we can have it is that we have a single-stranded messenger RNA that has folded back on itself, and the complementary pairing has caused it to end up as a double-stranded molecule. And those are the two ways in which we can end up with double-stranded RNA. They should not naturally occur. This is something going wrong in the system. Using the images, what you've got to do is try and come up with the stages that are involved in which the double-stranded DNA results in small interfering RNAs and the effect they can have with translation. So pause the presentation, use the images and words on the screen, work your way through, and when you think you've covered all the steps, you can restart the presentation and check how you've got on. So let's have a look at how small interfering RNAs can interfere with the process of translation. To start with, we have to get our double-stranded RNA and break it up into smaller parts. And that normally occurs with an enzyme. For example, in this case, they've told us it's dicer enzyme. The next thing that has to happen is that we have the two strands of the siRNA, and it has to associate with a protein, and when it associates with a protein it forms what is called an RISC, which is an RNA-induced silencing complex. You'll notice at that point the double-stranded siRNAs have separated out. The hydrogen bonds are no longer holding the two complementary sections together. What that means is that that small interfering RNA is now able to complementary bind with something else, and what it will complementary bind with is another messenger RNA strand that was on its way to the ribosome to be replicated. When it does that, the enzyme, the protein that's associated, cuts that messenger RNA up into sections, and as a consequence, the messenger RNA is no longer correctly transcribed into a polypeptide because we've essentially cleaved the gene at some point along the messenger RNA strand. So when it goes to the ribosome, it does not form a fully functioning polypeptide. Because the gene is no longer expressed, because we've interfered with the translation process, transcription occurs as normal, producing a messenger RNA. It's the translation we're targeting, and we say that the gene is no longer expressed and has become silenced. Well, here's a question. Are small interfering RNAs a bad thing? Well, the answer is, of course, they're not always. We can actually put them to use. Because we know what they do, we can actually put them to specific functions. And we can use them to target cancer cells and switch off certain messenger RNA and stop replication in cancer cells. We can also switch off other genes as well, so that certain phenotypes, for example, certain skin discolorations don't appear. And what we can also do with it is we use it to target some very specific diseases that we've associated with certain alleles in certain genes producing certain proteins and chemicals. If we know that these proteins and chemicals are being produced cause certain diseases, we can use these small interfering RNAs to stop the messenger RNA ever becoming a polypeptide or being used to make polypeptides, and therefore treat certain diseases. Just be aware in the exam that dicer enzyme does go by other names. The endoribonuclease dicer is not so hard to spot because it's obviously got dicer in it, but you can also talk about helicase, RNAs motif. What you're essentially looking for is an enzyme that chops up double-stranded RNA. If it's chopping up double-stranded RNA, the chances are that it's dicer enzyme. What we'll have to be aware of is that these small interfering RNAs are about 21 to 25 bases long, and while they can be made in the lab for obviously the medical reasons we've just mentioned, they often occur naturally. And if they occur naturally, they can of course cause huge amounts of problems in terms of phenotypes and gene expression for organisms. So from this presentation, there are two things you should be able to now do. You should be able to state what small interfering RNAs are, and you should also be able to explain how they're formed and how they affect gene expression, specifically how they interact with the process of translation.